ask them later. If nobody has anything right now, we're going to go ahead and we're going to start looking at our next topic, which is kind of a weird topic for a biology class, because our next topic, we're going to talk about viruses and viroids and prions. And the reason that this is kind of an unusual topic for this class is biology is the study of life, and viruses and viroids and prions are non-living. They're not alive. However, they do affect living things. All three of them can cause diseases. And so that's why we're typically interested in them when we study biology. Now, the basic reason that they're not considered to be alive is, remember that one of the characteristics of living organisms is that living organisms are made up of cells. Some living organisms, like most bacteria and archaea, and some organisms called yeast that we haven't talked about yet, most living organisms, or a lot, I should say, there's just one cell. Every cell is a complete individual. But we have some living organisms like people that are multicellular, where there are a lot of cells in our body. In the case of people, give or take about 40 trillion. But viruses can do two things that living things can do, even though they don't have cells. Now, since they don't have cells, that means they don't carry out any metabolic activities. They can't really respond to their environment. They can't do anything that requires some sort of chemical reaction, some sort of metabolic pathway. But they can do two things. Viruses can reproduce. But notice they can only reproduce when they're inside a living cell. Now, the reason they can only reproduce inside a living cell is when we look at viruses, they have two basic components. They have a nucleic acid. In some viruses, it's DNA. In some viruses, it's RNA. And they have protein. Well, just think about the protein part of a virus. For a cell to make a protein, it's got to have a stockpile of spare amino acids. It's got to have a ribosome to put those amino acids together to assemble them. It's got to have ATP because putting a protein together takes energy. Viruses contain no spare amino acids. They don't have any ribosomes. They don't have any ATP. Now, since these are metabolic reactions when we make a protein, that means they need enzymes to regulate them. Viruses typically don't have any enzymes to regulate making proteins. So viruses can reproduce, but they've got to be inside a living cell, because where are they going to get the spare amino acids, the enzymes, the ribosomes, the ATP? They're going to use the living cells. And when viruses reproduce, when they replicate their DNA or RNA, there can be mutations. And those mutations mean that viruses can evolve. So when we think about viruses, they really only do two things that we associate with living organisms. They reproduce and they can evolve. Some viruses evolve very quickly. I'm sure you've heard in the news about new strains of the SARS-CoV-2 virus, the UK variant, the South African variant, the Brazil variant. These different variants, these are mutations that have occurred. Beyond that, when we look at viruses, if you think about something like the flu virus mutates relatively quickly, which is why if you get a flu vaccine this winter, you'll have to get another flu vaccine next winter, because by the time next year comes around, the virus will have mutated. The vaccine that you get this year won't really be much help. So we'll talk a little bit later about how vaccines work, and we'll see why you might have to get a new vaccine. Right now, we all have our fingers crossed. The vaccines that have been developed against the SARS-CoV-2 virus seem to work against the variants that have evolved up to this time. So keep in mind, viruses are much smaller than cells. If we think about a cell, a virus is maybe the size of a ribosome. So even compared to a prokaryotic cell, they're small. Now, another way they're small is in terms of their genetic material. Just like in a eukaryotic cell or a prokaryotic cell, viruses contain genes. But we said the other day, a typical eukaryotic cell, like a human cell, might contain 23,000 genes. A prokaryotic cell, typically contains between about 5,000 and 10,000 genes. Now, remember, we said plasmids were much smaller pieces of DNA, maybe a dozen genes, maybe 100 tops. Think of viruses genetically as being about the same size as a plasmid. They might have really just 2 or 3 genes. They might have up to about 60 or 70. So 60 or 70 versus over 4,000 in a prokaryotic cell, over 20,000 in a eukaryotic cell, very small. All viruses have 2 parts. There are some other parts they may have, but not always. All viruses contain nucleic acid. Now notice that this nucleic acid can be either single-stranded or double-stranded. It's one or the other, they don't have both. And it can be either DNA or RNA. So if you remember from 105, one of the differences we talked about DNA and RNA in cells is that DNA in the nucleus, or in the nucleoid, if we're talking about a bacteria cell, DNA is double-stranded, RNA is single-stranded. There are some viruses that contain single-stranded DNA. There are some viruses that contain double-stranded RNA. But whether they have DNA, whether they have RNA, whether they have single-stranded or double-stranded DNA or RNA, the DNA and RNA can be divided into genes, just like we could divide the DNA in your nucleus into genes. And just like in your cells or prokaryotic cells, the genes are the instructions that tell the virus what's the primary structure of the proteins. So besides the nucleic acid, the only other part that all viruses have is something called the protein capsid. Sometimes just called a capsid, it's sometimes called a capsule. And this is essentially a protective layer that encloses the nucleic acid. Now, in some cases, the protein capsid looks like a container or a box, and then the nucleic acid is inside, except it has often much weirder shapes than just a box that you might get from Amazon or FedEx or UPS or whoever. A lot of times the protein capsid is shaped like, if you've ever seen any movies where they showed the old Apollo and Gemini and Mercury space capsules. Sometimes the protein capsid forms a structure that looks kind of like a tin can. So there are all sorts of different shapes. And we'll talk in a few moments, but the shape for the protein capsid is crucial in determining how the virus works. Now, there are some parts that other viruses have. Some viruses have some enzymes that they need to reproduce. If they've got some enzymes, they're typically found inside the capsid, very close to the DNA or RNA, to the nucleic acid. Now, remember earlier I said, why do viruses need to infect a living cell? Because they're going to steal enzymes from the cell. And that's true for the most part, but there are some viruses that need an enzyme to reproduce that eukaryotic cells don't have, or prokaryotic cells, because there are viruses that infect bacteria and archaea. So if they need an enzyme to reproduce that the cell that they're going to infect doesn't have, then they have to make that enzyme. So some viruses have enzymes, and some viruses have what's called a viral envelope, or sometimes just an envelope. And this envelope is a phospholipid bilayer. And this phospholipid bilayer is essentially stolen from the membrane of the host cell. Sometimes it comes from the plasma membrane. Sometimes it comes from one of the internal membrane systems like the Golgi apparatus or the endoplasmic reticulum. So let's look at a virus, one that's been in the news a lot lately. Let's look at SARS-CoV-2, which is the name of the virus. Notice COVID-19 is actually the name of the disease that this virus causes. But the virus itself is SARS. It's in a group of bacteria that cause severe acute respiratory syndrome, SARS. And it's a coronavirus, that's the CoV. So when we look at this, you can see all these little colored lines on the inside. These little colored lines are the nucleic acid. So this is the instructions to make the viral proteins. And in the case of this virus, we find proteins in 3 different regions. Notice that there's this blue squiggle that kind of curls around the nucleic acid. In the SARS-CoV-2 virus, this is the capsule. And when the capsule is tightly attached to the nucleic acid, like in this case, it's called nucleoprotein N. So this is showing, the SARS-CoV-2 virus, but notice that there are a lot of viruses that have nucleoprotein N. Notice also there's a typo in this picture. RNA, the N and the A aren't capitalized. So the SARS-CoV-2 virus is an example of a virus that has DNA as its genetic material. Now, if we look at the instructions that are on the RNA here, the instructions on this RNA make several different proteins. First of all, they make the proteins that are found in this blue squiggly capsid. So those are the instructions for those proteins. They're also the instructions for a protein called envelope small membrane protein E, which is some of these little kind of greenish circles sticking off to the inside. There are also the instructions for hemagglutinin esterase HE, these little green circles sticking off to the outside. And there are also the instructions for membrane protein M. So if we look at this circle made up of smaller ovals here, this represents the viral envelope. And this is a lipid bilayer, a phospholipid bilayer. It was taken from the cell that this virus infects. And then every once in a while, there are some little blue ovals. The blue ovals represent membrane protein M. And the protein that we're familiar with, because this is what all the pictures of SARS-CoV-2 show, is the glycoprotein spikes, which are referred to with the letter S. Notice glycoprotein tells us that these are a combination of carbohydrate, glyco, like in glycogen or glycolysis, and protein. That part's pretty obvious. Now, the glycoprotein is really crucial. And to think about why it's crucial, we have to look at the cycle of infection that a virus goes through and the cycle of reproduction. If you look this up online, a lot of places will call it the life cycle of the virus. But if you want to get technical, and we're getting technical in this class, it's not a life cycle because viruses aren't alive. You could call what humans go through from birth to death a life cycle, but viruses don't really have a life cycle. They're not alive. And I think partly because COVID-19 is such a serious illness, they've kind of relaxed some of the things that they used to enforce. If you see any advertisements for things like Lysol or Clorox, I've seen some advertisements where they say they kill 99% of all viruses. Well, nothing kills viruses. Viruses aren't alive. It's like lighting a pile of wood on fire and saying, this fire will kill 99% of all logs. Sorry. So let's look at the life cycle. And we can typically talk about 6 steps in the life cycle. We're going to come down and we're going to look at a diagram. So let me know if this diagram is too small and I'll enlarge it a little bit more. I think you can see it, but I'm not sure. So let's look how a virus infects a cell. And notice we would see a very similar pattern if we looked at any type of virus, any type of cell. It's a little bit different when we look at prokaryotic cells, but it still follows the same basic outline. So here's our virus, the SARS-CoV-2, and we can see those S glycoproteins, those spike proteins sticking out. Inside, we can just, we can see a little blue circle. That's our viral envelope, our double phospholipid layer. And then inside that, we have our capsid and our RNA. Well, if we look at SARS-CoV-2, for a virus to infect a cell, in most cases, that cell has to have a receptor that the virus can attach to. Now remember, receptors are molecules in the outer portion of the plasma membrane of a cell. And most receptors, not all of them, but most of them are proteins. And one of the things that we always want to stress about proteins is, how do proteins work? They work by their 3-dimensional space. They're very small, but they do have a specific shape, and that shape is how they work. When we talk about proteins working with other chemicals, we often talk about they work like a lock and a key. So they fit together with other chemicals very specifically. So the S glycoprotein, that spike protein, fits into this ACE2 receptor. Now, the ACE2 receptor is not here for the virus. ACE2 actually stands for angiotensin converting enzyme 2. And this is a chemical that's found in your blood that cells can pick up out of the blood. And so this ACE2 receptor is here for ACE, angiotensin converting enzyme 2. SARS-CoV-2 just happens to have a protein, that spike protein, that's similar enough in shape, it can attach. But notice that viruses can only attach to cells that have receptors they fit into. So this is why different types of viral infections infect different cells in your body. Viruses that cause colds can affect some cells in what we call your mucous membranes. So you might get a runny nose if you get a cold. You might start sneezing. You might get a cough. You might have watery eyes. But the viruses that cause colds can attach to cells in your heart. So they don't cause heart attacks. They can attach to cells in the brain, so they don't cause strokes. They can't attach to cells in your liver, so they don't cause liver damage. So viruses are very specific about the cells they infect. Now, once they attach, then the virus enters the cell. In the case of SARS-CoV-2, it actually enters still attached to the receptor. The receptor kind of pivots and twists and pulls this virus into the cell. Notice that in some viruses, once the virus is attached to the receptor, the envelope and the capsid break apart, and only the viral DNA, or RNA, as the case may be, only the nucleic acid enters the cell. But in the case of SARS-2, the whole virus enters the cell. So the virus has to attach, it has to enter the cell, and then once it enters the cell, the virus breaks apart and releases the nucleic acid. So this is showing our RNA. Now, in this case, the RNA that's found inside the SARS-CoV-2 virus doesn't actually have instructions for making the proteins for this virus. Instead, this RNA is complementary to the RNA that has the instructions. So the RNA that comes out of the virus is called a plus strand, and we make a negative strand by complementary base pairing. Now, this negative strand is used for transcription. So remember, in transcription, we take DNA, we make messenger RNA from it. You might think, since this virus has RNA as its genetic material, we could go straight to making proteins, but that's not how it works. We've got to make this negative strand of RNA. Now, once we have this negative strand of RNA, this negative strand of RNA gets used for two different processes. It gets used for replication. So in other words, the cell makes more copies of this messenger RNA. Well, if for the virus to reproduce, every new virus that's produced has to get one copy of this RNA. The other thing that this RNA negative strand is used for is it's used for transcription. It's used to get the instructions to make all those proteins that we talked about before. The spike protein, the N nucleoprotein, the M protein, all those proteins we make the instructions for down here. Now, again, remember what I said, the virus can't do this without the cell. So these pieces of messenger RNA that get made from the negative strand of RNA up here, these pieces of messenger RNA are transferred to ribosomes on the rough endoplasmic reticulum. And those ribosomes carry out translation and use the information on this messenger RNA to make the proteins that are found in the virus. Then, and don't worry about some of these names, every stage of this process has a name. These proteins that get made in the ribosomes are modified in the Golgi apparatus. So remember, we talked about that when we discussed protein synthesis in eukaryotic cells. And then when the proteins come off the Golgi apparatus, remember that proteins come off of the Golgi apparatus in little membrane sacs called vesicles. So these vesicles, which have two layers of phospholipids, these sacs pinch off the membrane and the proteins start to be assembled to make the complete virus. So we have all these proteins pinching off inside a vesicle, and that vesicle is going to be what makes the virus. Then we ourselves put the virus together, and the virus is released from the host cell, the cell that it's infected. Notice this chloroquine with all the lines here. You might remember this is one of the chemicals that was proposed as being a potential treatment for COVID-19 because under various circumstances, chloroquine can block some of these processes. Notice that the little kind of blocking symbols are coming out to various arrows representing metabolic pathways. Now, it turns out, as far as we can tell, chloroquine doesn't really do anything in this instance. But you might remember it because President Trump proposed this and some of its relatives as being a treatment for COVID-19 way back in the beginning of last summer, I think, or the end of last spring. Now, again, notice there are some differences. I mentioned when a virus enters a cell, the whole virus may enter, as in the case for SARS-CoV-2, or only the nucleic acid can enter. When a virus leaves a cell, Some viruses leave a cell very gently and the cell stays alive and continues producing viruses. Some cells, when they leave the cell, they pop a hole in the plasma membrane. And if that's the case, then that kills the cell. There are different ways, there are different alternatives for some of these things that we're talking about. So let's look at how your immune system fights off an infection, like if you get COVID-19. And notice what we're talking about here, even though we're focusing on viruses, also applies to bacteria and to fungal infections like ringworm or athlete's foot. Ringworm isn't caused by a worm, it's caused by a fungus. And it's also caused the same way that your immune system fights parasitic worms and things like that that might get into your body. So we have a couple of types of cells in your immune system that do different things. So one of the things we have is some cells called macrophages and neutrophils. And macrophages and neutrophils phagocytize viruses or bacteria or other organisms. And they do this by endocytosis. Remember, endocytosis was a process where we brought a large chunk of material into a cell. So here, the large chunk of material is the virus. And once the virus is brought into the cell, remember that it's enclosed in a little membrane sac. That sac is then fused with a lysosome. Remember that lysosomes contain digestive enzymes. And so the virus or the bacteria or whatever is destroyed in this fashion. Now, notice that sometimes macrophages and neutrophils don't only destroy viruses, but going back to our diagram up here, if a cell is infected by virus, sometimes these macrophages and neutrophils can detect that there are viruses entering the cell. And so sometimes some of your immune system cells actually destroy your own cells. The idea is kind of like, you know, the old movies about World War II, where the hero throws himself on the hand grenade that lands by his friends. The idea here is your immune system decides, okay, it's better to sacrifice this infected cell than to allow it to make more viruses. And actually the infected cell starts sending out chemical signals that attract immune system cells and that also warn other cells in the area, hey, there's a virus around, you better start preparing to be attacked. The two cells that do most of the work in an infection, though, are some cells called B cells. And B cells are made and mature in your bone marrow, so the inside of your bones. Eventually, when they're fully mature, they make some things called antibodies. And what are antibodies? What else? They're proteins. These are the proteins that attach to antigens. And antigens are almost always proteins too. So what's an antigen on the SARS-CoV-2 virus? Well, it's some of these molecules that are sticking out on the edge, where a cell of your immune system can recognize that cell. And so the one that your immune system responds to on the SARS-CoV-2 virus, the one it responds to most strongly is these spike proteins, the ones that stick out the farthest. So basically, your B cells make antibodies. And antibodies are kind of like grappling hooks, I guess you could think of them. They attach to these antigens, so to these spike proteins. And when they do this, notice they're changing the shape of that protein, which means that that virus or bacteria or whatever it is going to have a hard time getting into your cells. And eventually, other cells like macrophages can come along and they can say, oh, here's something that some antibodies are attached to. That's something we need to destroy. Now, Notice that when you get an infection, you make antibodies. This is how you fight off the infection. So after you've had an infection, so if you've had COVID-19, one of the ways that we can tell this, that you've probably heard of, is we can do an antibody test. We can take a blood sample, and if you've had COVID-19, you still have antibodies circulating in your blood. Now, our best evidence so far is that if you've had COVID-19, these antibodies continue to circulate in your blood for about 5 months, but they gradually decrease in number. So it's after you have COVID-19, you probably have some pretty good immunity for 5 months, but at the end of 5 months, probably you're susceptible again. And there have been several cases that we know of where people have gotten COVID-19 2 times. How can that happen? Because your immune system after a while doesn't respond as strongly. Now we'll talk about that again in a moment. The other type of cells that respond are something called T cells. And T cells mature in your thymus gland, which is in the front of your throat. And these are the cells that destroy your own cells that are damaged and can't be salvaged. And they're also the cells that kind of manage the overall response. Now, when I say they manage the overall response, they recruit other cells to attack these foreign antigens that are in your body. Now, notice when we think of foreign antigens, your immune system evolved to attack bacteria, viruses, fungal infections, parasitic worms. But it can also attack foreign cells that we want in your body. If you get a heart transplant, your immune system doesn't recognize those new heart cells. If you get my heart, your recognition proteins on your heart cells are different than the recognition proteins on my heart cells. So when your immune system encounters this heart that you just received in a transplant, your immune system's response is, these are foreign cells, we need to get rid of them. So if you know anyone who's ever had a transplant, not just a heart transplant, but any type of transplant, one of the things that they usually give people who have had a transplant is what are called immunosuppressant drugs. And immunosuppressant drugs suppress the immune system. They reduce its reaction. So for people who have had a transplant, usually you can't go visit them right after the transplant if you have any type of infection. Why? Because if they get this infection, their immune system isn't responding very well. So it might be a pretty minor infection, but they could have serious problems. Now, notice that some of the T cells and some of the B cells end up forming what we call memory cells. And these cells can stay in your body sometimes for years. Depends on how strong your immune response was and your immune system. It depends on a couple of factors. Some of these memory cells stay in your body for decades. Some of them are gone in a few years or a few months. But as long as the memory cells are there, this means the next time you get exposed to that virus, to that antigen that caused your first response, the second time you get exposed, your immune system responds faster because it already knows what this antibody looks like, how to recognize it. And not only does it respond faster, it's a stronger response because your immune system knows this antibody shouldn't be here. And it's a longer lasting response. So this is why in most cases, the second time you get exposed to the same antibody, you don't necessarily develop any symptoms. So there's a childhood illness called the mumps. And if you get the mumps, and it's an infection in the tonsils, if you get the mumps, the next time the mumps goes around your school, you don't get the mumps usually. Why? Because you have memory cells that respond to this infection, and as a result, you never even know you were exposed. Now, memory cells do gradually decrease over time. Sometimes it takes decades, sometimes years, sometimes just a few months. And so this is why for some vaccines, it's recommended that you get what's sometimes called a booster shot. You're supposed to get a booster shot against tetanus every couple of years. Why? Because the memory cells against tetanus, and we don't know exactly why in terms of, why some diseases, the memory cells last longer than other, but for tetanus, the memory cells just don't last that long. So you have to get, in a sense, revaccinated. So when we look at viruses, viruses aren't alive, but because they infect living cells, that's why we study them in biology. Now, keep in mind, if we look at the human nucleus, about 8 or 9% of the DNA in the human nucleus is actually DNA that comes from viruses. So remember when we looked at bacteria the other day and we were looking at a virus transferring DNA from one bacteria cell to another, we saw that a virus can integrate its DNA into the host DNA. It can stick its DNA into the host chromosome in a bacteria. There are viruses that can do the same thing in eukaryotic cells. So if we look at you or me or anybody else, about 8 to 9% of our DNA comes from viruses. Now, these viruses must have infected people a long time ago because my viral DNA, if we look at it, is going to be almost exactly the same as yours. Since different groups of people have, evolved in different areas over the years, there can be some minor differences, but they really are pretty minor. So now let's think about viroids. Whenever we see oid at the end of a word, oid means like or similar to. The nucleoid is like a nucleus. How is it like a nucleus? Well, mostly because it has a chromosome with DNA, and the DNA makes up genes. So viroids are kind of like viruses in a way, except you could think of them as being a part of a virus. A viroid is just a small circular molecule of RNA. All the viroids that have been discovered infect plants. It's sometimes called a small circular molecule of naked DNA, RNA. And naked just refers to the fact that the RNA is out there all by itself. It's not attached to anything else. And what happens is when these viroids enter a plant cell, they attach to DNA in the nucleus. And then that interferes with protein synthesis. If you think about it, that would interfere with the transcription stage of protein synthesis. So when viroids do this, they cause some plant diseases. Now, we don't hear a lot about these plant diseases because they don't affect people. They only affect plants. But some of them actually do have some important effects on agriculture by killing a lot of plants. For instance, there's something called potato spindle tuber disease. Potatoes are a type of plant part called a tuber. And in spindle tuber disease, the potatoes get kind of long and skinny. Not exactly like a French fry, but kind of more that shape than the typical kind of oval or roundish shape that we associate with potatoes. Now you might think, why does this same type of viroid affect potatoes and tomatoes? But keep in mind, potatoes and tomatoes, even though they may not look real similar, are actually pretty closely related. They're in the same clade of plants. So it's not real surprising in that sense that a viroid that affects one would affect the other. And then finally, let's look at the last category, prions. Now, I said today, and I know in 105, you probably got tired of hearing me or whoever your teacher was say that when we look at proteins on a molecular level, everything works by its three-dimensional shape. Prions are proteins, and they're folded into an abnormal three-dimensional shape. And, excuse me, what happens is when prions get into a cell, they can cause other molecules of the same protein to misfold and change their shape. And all these abnormally folded proteins end up forming a clump. And this clump is generally in the brain because all the prions that have been discovered so far affect the nervous system, and they affect the nervous system of mammals. We haven't discovered any that infect, insects or something like that. But it may just be because we don't really care if insects have brain damage. I'm not sure how closely anybody has studied that. And these amyloids cause cell death. So they cause the death of cells in the brain. The best known prion infection is something called bovine spongiform encephalopathy, or BSE, which is what's commonly called mad cow's disease. In mad cow's disease, brain cells in the cow are being destroyed, and the cows gradually start having problems. One of the first symptoms we can see is because some of the cells in the brain that tend to get destroyed very early are cells that coordinate movement. Cows that have this may start having trouble standing up or walking. They may fall over. Now, the problem with cows that have mad cow disease is if you happen to eat one of these cows, that means that you could potentially get this disease. And there have been several cases where mad cow's disease has been transmitted to a person. Now notice, this prion infects brain cells. So most people don't eat brain cells, but as the cells become damaged, there can be small amounts of these prions circulating. So usually when we discover that some cattle have mad cow's disease, all the cattle are killed and their carcasses are burned. Because keep in mind, what we're talking about is an individual protein molecule. So when we look at this, it's kind of like when we talked about viruses a few moments ago. You can't really kill an individual protein molecule. So high heat can destroy it. Remember back in Biology 105, we talked about denaturing proteins, but we can't necessarily kill it. So you can't cook a cow that has mad cow disease and say, oh, it's safe to eat now. And actually the protein that causes mad cow disease is represented in this diagram. And this diagram is a way of showing what we call the secondary structure of a protein. Now you might remember that the secondary structure of a protein is what happens when we have amino acids that are right next to each other and they start attracting each other by forming hydrogen bonds, or they start repelling each other because one of them is polar and one of the R groups is nonpolar. The protein on the left is actually the normal protein. And if we look, there are 4 different areas. 2 of them are shown in kind of a yellow gold. 2 of them are shown in gray. And these areas all have an alpha helix shape. Remember, that's the basic shape that you see in a spring or a slinky. And we said that was one of the basic secondary structures that we see in proteins. Now notice that these alpha helix areas are connected by some other regions. This is an area of the secondary structure that's sometimes called a turn or a random coil. This is the connection between one section of alpha helix and another. Because of the mutation that occurs when we look at the mutated protein, the one that is the prion, what we see is we still have our two gray alpha helixes, but besides those two gray alpha helixes, we have two areas in here that are beta sheets. Remember, the beta sheet was another secondary structure. And notice that even our alpha helixes, although they're here, they're oriented in a different direction. They're not going in the same direction. And if we notice, the shape of these connecting sections is also different. So this changes the shape of the protein. Now, notice that some people get a disease called Creutzfeldt-Jakob disease. And this is a genetic disease, so this is inherited. But in Creutzfeldt-Jakob disease, you actually inherit the mutation that causes the protein to misfold. So you could get this illness by eating a cow with it. You could get this illness through inheritance if it runs in your family. If you have this, it sometimes causes a disease called fatal familial insomnia. So I'm sure you all know insomnia is when you can't sleep. This is a type of insomnia that's genetic, so it runs in families. So that's the middle part of the name. And it's such a severe insomnia, people who have it eventually die from lack of sleep. Now, we don't know exactly why you need sleep, but we do know that, some people need less sleep than others, but people typically have to sleep at least some amount to survive. And in this case, people don't sleep enough to survive. It also can cause, the same prion, can cause a disease called Kuru. And Kuru was first discovered when the Europeans started exploring the world. When some of the Europeans were exploring New Guinea, an island down near Australia, at the time, New Guinea was inhabited by a lot of small tribes. And kind of like the North Americans, these small tribes were constantly battling with neighboring tribes. They were pretty low-key battles in terms of, injuries and deaths. They weren't big scale wars or anything like that. But here was the problem. Every once in a while, one of these tribes would kill one of their enemies in battle. And like a lot of ancient religions, one of the beliefs that most of these tribes shared was, if you killed an enemy, you would like to somehow absorb this enemy's strength. So how do you absorb this enemy's strength? Well, by eating them, of course. So that most of the tribes practiced cannibalism, at least occasionally. Now, when the Europeans came to New Guinea, one of the things they discovered was this disease called Kuru, which was basically a type of insanity. It made people crazy, but they really only found Kuru in the chiefs and in the shamans, the religious leaders of these tribes. Why would you only find it in the chiefs and the shamans? Well, it turned out, as people began to study the cultures more, when you killed an enemy, the warriors in the tribe would eat some of the muscle. But the brain was considered to be, important in terms of thought. So if you wanted to gain your enemy's wisdom and intelligence, what would you eat? Part of their brain. And that was what the chiefs and the shamans ate, was part of the brain. So they tended to get this disease and no one else did. So when we look at prions, notice that if we look at Alzheimer's disease or Parkinson's disease or ALS, amyotrophic lateral sclerosis, sometimes called Lou Gehrig's disease, these are diseases that are all caused by amyloid plaques, those clumps of protein. Now, right now, there's no evidence that any of these are caused by prions. I want to emphasize that. But it at least suggests that maybe prions have something to do with this. And maybe if we learn how prions really work, we can also learn how these diseases really work. Now, there is a problem, which is right now for any diseases caused by prions, there aren't any cures. There aren't any treatments. So this is why when some cattle get this disease, we burn the cattle, we destroy the carcasses, because we can't have them sitting around. It's too big a risk. So in England one year, they discovered some cattle with mad cow disease, and I think they eventually ended up having to kill more than 1 million cattle. So may have kept the country safe from disease, but think of the economic impact on all these farmers who, you know, put all this money into these cattle, and all of a sudden you had nothing to show for it. So when we look at viruses, viroids, prions, notice they all have something in common. And what they have in common is they're not really living because they're not made of cells, but they can all get into cells and reproduce. And they can mutate if the DNA, or in some cases RNA, that is the instructions, changes. So they can mutate and evolve potentially. Now, keep in mind, 50 or 60 years ago, if you would take a biology class, you know, we thought there was a very clear line between cells, which are alive, and things that aren't cells. They're not alive. You know, it's a clear division. However, what these three types of things show us is maybe the line isn't quite as sharp as we might think. Because think about it. Prions can reproduce, but all they are is a single protein molecule. Viroids can reproduce, all they are is a single RNA molecule. And normally we say, you know, individual protein molecules aren't alive. Individual RNA molecules aren't alive. Okay, but these guys can reproduce. They can do certain things that living organisms can do. So where do we draw the line becomes part of the question. Are there any questions about viruses or viroids or prions or anything else that we've talked about or just that you're curious about? Well, if there aren't any questions, I'll let you go for the weekend. And looks like it's going to be nice and sunny, just a little bit chilly. So on Monday, I'll tell you why it's going to be colder if the skies are clear. So enjoy the weekend. You can think about that.
Mr. Griffith?
Mm-hmm.
Yeah, I have a concern concerning the one bag equation.
Mm-hmm.
Yeah, there was, I think we used, so the time we used an equation like that, we're using x squared equals, okay, x squared plus 2pq, okay, p squared plus 2pq
Plus q squared.
Q squared. Yeah. And we're saying that it's going to be complex if we use more than two
Alleles.
Alleles. Yeah. If we're using 3 alleles and we're trying to use like binomial theorem, like trying to expand it using binomial theorem, I think can't
Well,
be
and
easy
that's really why so far all the examples that we've worked, we've stuck to just 2 alleles, because the mathematics gets much more complicated. Depending on what we have measured, we could solve it if we put some numbers into a computer. But it would be numbers that if we were to work it out, instead of, you know, taking a few minutes to work the problems like we did in class, just to answer one of those questions would probably take you an hour or 2. So we'll let the computers handle that. Kind of like back in 105, we did monohybrid crosses and dihybrid crosses, but we didn't do a trihybrid cross.
Right.
And we could do it, but if you do a trihybrid cross, your Punnett square can be 64 cells. So it would really take forever.
Okay.
So
thank you.
That's what computers are for, repetitive work.
All right. Thank you.
You're welcome. Have a good weekend.
You too.
Have a good weekend.
You too, Nick.
Bye.
Bye-bye, Maggie.
Have a good weekend.
You too, Jade.
Have a good weekend, sir.